Thank you for the introduction and the invitation in general. This is going to be a really interesting conference. So today I'm going to talk about some research we've been doing on streaming approaches for dealing with the sequencing data crush. I'm going to start with some acknowledgments, because that way I don't have to worry about rushing through them at the end. Talk about some of our approaches for big data and next-gen sequence analysis. And then by invitation, I'm going to make some sweeping generalizations about biology and physics and point out some of the differences between the two disciplines. And we'll see how that goes. So just in terms of acknowledgments, a bunch of people have contributed to this work, including some collaborators out here at LBNL and JGI, and a bunch of different graduate students. And I go over this in my blog post, which, well, actually, just would like to point out I'm here. I'm sacrificing time with two of the most attractive girl children in existence, and my wife is currently taking care of them, so she's also very important for me being here. So I hope you appreciate the sacrifices I have to make in order to come talk to you. Okay, so we practice open science, which is not necessarily normal in biology. There's a blog post. If you just type Titus Brown blog, you can see links accompanying the slides, the talk itself, et cetera, et cetera, et cetera. So what I'm going to talk about today is not actually a problem motivated by healthcare. And it's a different big data problem, which is actually the problem that there's this vast microbial, the vast microbial world out there about which we know virtually nothing, and in particular soil. So being from the Midwest, we have lots of soil. Soil is full of uncultured microbes. It's actually incredibly important in terms of carbon and nitrogen cycling, as well as all sorts of other things. You've probably all seen the Human Microbiome Project stuff that's been coming out over the last couple months. And that's another aspect of microbial ecology and microbiology that we're just starting to get a handle on, mainly because until recently we didn't have the sequencing capacity or the investigative technologies to do a good job of looking at what's in the environment. So if you just go out and you start sampling from different soil environments, these are from 9 different Midwestern samples, so Iowa, Kansas, and Wisconsin, and you just start looking at species tags using sequencing of uncultivated microbial populations from soil, what you find is that there's at least thousands and perhaps millions of species in each gram of soil. So we sampled 9 different grams of soil and then asked, hey, if we sample 10,000 different sequences that are from a particular gene that's known as a species tag called 16S, do we run out of things to sample at some point? And the answer is no. If you only go out to about 10,000, you find that, whoops, if you only go out to about 10,000 samples sampled randomly from the environment, you find that you're still picking up. The slope does not go to 0. You don't saturate this curve, and we're still picking up quite a few novel species, even after we've seen 1,000 to 1,500 different species. So our suspicion, incidentally, and I provide some links in the blog post for this, is that there's upwards of 1 million different species in each gram of soil. So about 10 million different bacteria, 1 million different species. And these are mediating a lot of environmental processes in which we are very interested for reasons that are different from illness, but equally important, I think, on the global stage. So the big shift in biology over the last maybe 5 to 10 years is that it has become easier to pick up biological samples and feed them into a sequencer and generate a lot of computational data than it is to do pretty much anything else in biology. You can go out and as long as you can get DNA from a sample, you can sequence it. And the sequencing, and this is very important, the sequencing happens at random. This is shotgun genomics or shotgun metagenomics in this case. And essentially, you're reaching into the population of genomes and the population of microbes, and you're picking at random from them. And most of the genomes that we have today have been sequenced this way. And the key component here, of course, is that you generate all this data, and then you give it to somebody that can run it on a computer and give you back a genome. So the computational task at hand is to take, the way I like to characterize the problem is, suppose you take a bunch of different libraries and you feed them all into a paper shredder, and your task is to reassemble the original texts in those libraries from the pieces of paper you get out. And one point here is that imagine that you throw away about half those pieces of paper, so you're really relying on the redundancy present in the text in the library to actually reassemble the books. And it's an immense problem, and what makes it even more fun is that the sequencing is error-prone. So when you get out these sentence fragments from the paper shredder, they may have errors in them in the process of turning them into computational data. And so your job as a computational biologist working on assembly is to recover this original text from this very messy, error-prone data set. And so because we're sampling at random, coverage becomes very important. And so by coverage, I mean how many original copies did you have in the sample that you sequenced? Or how deeply did you sequence a sample? So if you feed in, say, one copy of A Tale of Two Cities into a paper shredder and throw half of the paper shreds away, in other words, you cover only to about half-X coverage of the book, that means you can't actually reassemble most of the book, right? You're missing half the data. Because the sampling is random, it turns out you actually need to go out to an average coverage of maybe between 10 and 100 to actually be able to reassemble a genome from scratch using this random sampling, using data generated from shotgun sequencing. And we'll come back to this in a sec. So the dominant assembly paradigm at the moment is something called De Bruijn graphs. You essentially break down your sequences into shorter k-mers, build overlap graphs of those k-mers, and then you can read off with an Eulerian search, you can read off your assembled sequence. There's various reasons you can read this excellent review. It's why this is actually more efficient than some of the other approaches, but it is basically the dominant approach. And the big problem with it is that as soon as you put in errors, you start generating bubbles in the graph where this G, which should be a T but was read erroneously, the error rates are in the 0.1 to 1% range, so about up to one error per 100 characters. You get these big bubbles in the graph that don't rejoin quickly, and a lot of effort has been put into figuring out how to deal with this problem, because we're talking about billions and billions of sequences here, potentially. So in particular for these De Bruijn graph approaches that lead to assembly, you end up with the problem that as you sequence, as you generate more and more data, you generate more and more errors, which start to dominate the De Bruijn graph and increase the memory required for doing assembly linearly with the amount of data you have. So even if your true information in your sample saturates, the deeper and deeper you sequence, the more errors you get, and the bigger a problem this becomes. And unlike many of the problems that you see in sort of the O'Reilly Scala is it? It's Strata, sorry. Strata conference. This is really a situation where you have a big graph. It's a big memory problem because you load that graph into memory. There's not very much in the way of locality in the graph. So you can't really distribute it that easily. You have to come up with clever ways to do that, which in general haven't been done. And essentially, you need terabyte memory machines, and I'll go into some of those requirements in a sec. So it's a very annoying problem. OK, so I was glad to see Jacek put up some numbers. So I estimate the worldwide capacity for DNA sequencing to be about 15 petabases per year. That's based on public estimates from Illumina of the number of machines they've sold. It's probably quite a bit larger. Individual labs can generate 100 gigabases in 1 week for about $10,000. And the reason I like to point this out is it's not like we have one company or 5 centers around the world that are doing this sequencing. Each individual biology lab is writing NSF grants, getting money to do this, sending it to the sequencer, getting back the data, and then going, oh no, what now? Right? Because most biology labs don't have anybody that can deal with this data. They don't have any training in it. The software packages all run on this thing called Unix, which you may have heard of. And it's just very tough. So the sequencing's done at a boutique level. The sequencing formats are boring and semi-standard, but the basic analysis approaches are only about 80% cookbook. And so there's a lot of educational stuff that I'm doing that to try and train graduate students and postdocs in this problem, because of course biologists generally receive no training whatsoever in computation. And I'd also like to point out computational people generally don't know anything about biology either, which turns out to sort of block communication. And I'd like to point out this is probably the wrong group to make this point, but a lot of the technology that's being produced is being optimized for high performance rather than high throughput. And we don't need high performance as much for some of this stuff as we do high throughput. And I'll be really interested in hearing what tomorrow's sessions have to say on that. So my problems are also very annoying. I don't represent all of biology, just one very small subset of it. And basically, they're on the petabyte scale, because if you want to go out and you want to do a proper sampling of multiple grams of soil to figure out what the metabolic capacity of the soil is, you probably need to generate a couple petabytes in terms of proper samples. So we need about 50 terabase pairs of sequence for each gram of soil to fully sample what we can already observe as being there. By the time you do a statistically sound sample across a spatial grid, you're really talking about petabytes worth of sequence. This is, by the way, something that an individual lab might be generating in 2 to 3 years with the pace of technology. And our estimate right now is, using the current techniques and algorithms, is you need about 500 terabytes of memory on a single computer in order to assemble this volume of sequence. We don't have that much sequence yet, but we will. So what do? So this has really driven my lab's research over the last couple of years. So the two bits of technology that I'll talk about, and this stuff's actually published, and the next one is in preprint form, deal with the problem of doing compressible computing, in effect, on this data, which is really what you want to do. The information content of these sequences isn't actually as ridiculously high as it looks, because a lot of the, quote, information is actually errors, right? This is a standard problem with noisy measurements. You go out, you measure a lot of stuff, and you want to distinguish signal from noise. And so we've been investing a lot of time and effort in compressible computing, which essentially aims to do the computation on the actual information content rather than on the information content plus all the errors. So the two approaches we've developed, we've actually developed a couple more, but the two that I'll talk about today, one is compressible de Bruijn graphs. And I'm sort of dangling the raw meat out in front of the physicists in the audience here, because there's some pretty cool stuff going on with percolation theory, which I know everybody likes to talk about. So the problem with de Bruijn graphs is they're expensive to represent. Each of the nodes in the graph represents a 14 mer, and then the links between each node are 13 mer overlaps. And storing these in main memory is very tough, so we developed a probabilistic representation for de Bruijn graphs by using something called a Bloom filter, which I'm going to assume everybody's either familiar with or can look up in Wikipedia. The key point about a Bloom filter, which is basically a hash table without collision tracking, is that you can resize them. You can make small Bloom filters that have a lot of collisions and large Bloom filters that have a low number of collisions, essentially a set membership data structure. And so we figured out a way to store de Bruijn graphs and traverse them in a Bloom filter, which lets us take graphs and compress them down by decreasing or increasing the size of the underlying Bloom filter, at the expense of bringing in false positive nodes and edges. And so we end up with these graphs where you have the real connections between sequences, which are the things that you want to traverse out, and then you have false edges between nodes. And the question is, how far down can you compress the graph before these false edges start to cause trouble? So just to give you a little bit of intuition, I often speak about this to computer scientists and biologists. Suppose you have a map, a very big map on a piece of paper, and the map's really too big for you to look at in one step. So what you want to do is compress it down a little bit. So with the Bloom filter approach, what we're essentially doing is crinkling the graph at a local level, essentially embedding the graph in a lower dimensional space, kind of. It's not a perfect analogy. You're crinkling the graph, and you want to know how far can I crinkle the graph at a local level before the graph the structure of the graph, the structure of the map starts to become problematic, before I want to figure out how to get from here to here, and I see erroneous roads that don't actually exist on the real map or in reality, but just because of the compression of the graph, they exist. They look like they exist. How far down can we go? And the answer is actually pretty far. We can go down to about 18% false positive rates in the Bloom filter. And what you can see here, because the Bloom filter only introduces false positives, what you see is you can start with a circular graph. You can compress it by storing it in increasingly small Bloom filters. And you end up elaborating the graph at a local level, but up to about 18% compression rates, which you don't actually introduce false structure at a larger degree. And I took out some of the details, but essentially we looked at things like graph diameter. Do you start to be able to take shortcuts across the graph because of your compression? So the cool thing, and this was sort of an offhanded comment I made to the graduate student involved, Jason Pell, was, hey, if we can tie this to percolation theory, about which I knew basically nothing, then we can show that what we might be able to show is that you can go from a sudden transition, a phase transition, from an unconnected graph to a completely connected graph, which would mean that up to that point of compression, if we can figure out where that is, we're good, right? And so we were able to show exactly that. If you look at the fraction of active vertices, you put this in a bond percolation model, which has the convenient fact of not being analytically solvable, right? So we didn't have to worry about the hard math. We just had to run simulations. You see that essentially the component size undergoes a discontinuous transition at around 18% false positive. And we have all sorts of data showing that this is what's going on. And so what we're able to show is that for sparse graphs, at various compression rates, we get good graph representations for overall connectivity of the graph, and it's considerably better than the best possible information theoretic or entropic encoding, which is pretty cool. So for sparse graphs, we can go 2 to 10 times better than the best possible actual implementation of an exact de Bruijn graph. Now, when I say best possible, these do not include implementation details, right? So we're not saying these would be fast to access or anything. Just say that if you want to store all the information not using a probabilistic data structure, you're in trouble to this extent. It has a couple other nice features that are useful for biology. So on top of this, what we did was implement graph partitioning. Because we're looking at microbes, we're looking at many millions of microbial species potentially. We actually, when you look at this assembly graph, which represents connections within the actual genome, we can pull that graph apart into separate components. We can essentially execute a divide and conquer approach, which lets us break out all of the different components in the graph, all of the sequencing reads that belong to the different components, the different species in the graph, into different bins and essentially assemble them in massively parallel. So we've actually implemented this. It works extraordinarily well. It's incredibly convenient. It actually seems like the graph theoretic component distribution represents individual species, which is sort of ultra cool, right? How often do you have a neat theoretical idea that actually works out directly? It let us scale about 20-fold, about 20x over current approaches, which wasn't enough because I didn't put the graph in. I probably should have, but we're essentially, if you wait a year, the Moore's law capacity of computing is falling well behind the greater than Moore's law capacity of sequencing. So spending 2 years to come up with a 20-fold better solution for assembly meant we were still well behind the curve. So that was depressing. So the other point the other approach that we used is to develop an online streaming lossy compression approach. And I would like to point out that one of the things I really dislike about biologists is they don't understand what any of these words mean. And so when I go and give talks and I try and point out how awesome it is not to have to look at any piece of data more than once, they sort of give me a blank look of incomprehension. And they don't recognize how super cool this approach truly is. So I'm hoping for a good reaction here, okay? So the observation is when you look at the sampling distribution, the number of times each base has been sequenced, suppose you know the book you're sampling, and you look at the number of times each base has been sequenced, in order to sample every base, every component of that book more than 0 times so that you could at least in theory assemble it, you need to sample most of the stuff many more times than once, right? And so everything to the right of this blue arrow is massively redundant. And in fact, it turns out you don't even need anything greater than 5x coverage were it systematic in order to assemble a genome. So essentially, most of the data you collect from shotgun sequencing is redundant. And this holds true for resequencing analysis. This holds true for de novo sequencing. This holds true for mRNA-seq. This holds true for really pretty much anything that you're doing with Illumina sequencing. So what we did was we observed in particular that if you had a mixed population of 2 species, A and B, and species A was 10 times more abundant than species B, and you reached in and you sampled at random, and you wanted to recover species B from the population, you would be oversampling species A by a factor of about, 81% of your data would be unnecessary. And because that data contains errors, and you need to load that data in to resolve those errors with current approaches, this data was not only redundant, not only occupied disk space, but occupied memory, which is our big problem. So what we figured out how to do was develop an online algorithm for building this de Bruijn graph and then downsample the reads on the de Bruijn graph where we had already seen enough data to cover that portion of the de Bruijn graph. So essentially, rather than actually doing an assembly, what we do is we load in the data until we find some data that's redundant, and then we throw that data out. And it's a super cool algorithm. It's single pass, and because we rely on a count min sketch, it's fixed memory. And I don't know how many people out here have algorithms that are single pass and fixed memory, but pat yourself on the back, right? Because they don't get any better than that, right? So we look at each read, at each sentence that we get out. We look at the estimated coverage by mapping it to the De Bruijn graph, which is actually very fast. We can actually saturate the I/O of our HPC with only 4 threads with this algorithm because the computation's so trivial. If the read is not redundant, we load it in. If the sentence that we're looking at is not redundant, we load it in and save it. Otherwise, we throw it away. And this has an incredibly profound effect on our ability to deal with the data. So we can go, and with simulated genomes, it works on real data too. But on simulated genomes, where you have, say, a 400 KB genome that you've sequenced to about 200,000x coverage, the true information, you have about 400,000 20mers in that data set, 400,000 words, 8 million words in the sequencing data set because of all the errors. And with a single pass approach, we can throw out 5/8 of that data, 80% of the data and 5/8 of the noise. And if you're willing to do a little bit more work, go over the data 3 times, you can actually throw out 95% of the data, keep almost all of the original data, losing only 4 k-mers out of 400,000, and keeping pretty much only the true ones. And this works extraordinarily well on pretty much every data set we've tried it on. And I should say this number is linearly related to memory. So we can actually now do most microbial genomes, most transcriptomes on laptops without any problem whatsoever. And we're talking dozens of gigabytes of data potentially being just discarded. So, okay, so back to the original problem, which is, oh my God, we have a lot of data coming from soil. What do we assemble? So we've been able to complete some of the first and largest assemblies ever done of genomes from soil. And we get, in terms of the total assembly, we get about human genome size amounts of data. So the amount of data going into this is about 300 gigabases. We get about 1 human genome worth of data and a couple million contigs, assembling only about 20% of the data because of low coverage issues. We get 5 to 7 million novel genes that nobody has ever seen before from each one of these. Each one of these represents about a $10,000 data set, by the way. And 400 to 600 novel species just in the small amount of data that we have. So this is 1/10 to 1/100th of the amount of data that we know we want to do. And already we're getting essentially, basically annoyingly large amounts of novelty. Yeah, so I can talk more I talk about this problem extensively in most of my biology talks, like, well, now what? We've got the assembly. What does it mean? We have no idea. So, right, there's an advantage. I'm in a computer science department, so that's actually an acceptable statement, right? Hey, I helped you with your computational problem. You guys got to do the biology. And so now I'm making the mistake of switching to the biology department as a focus, and now they expect me to actually learn something about biology from all this data, which is much more problematic. So just some quick concluding thoughts on this side of things. So we've gone from theoretical approaches that should work all the way to implementation at scale, all the way to actually executing this stuff on real data and obtaining real results. And this is some of the sexiest stuff I'm doing. We've done this on a lot of other problems where essentially our approach is let you discard 99% of your data, get the same answer out the other end, and do the computation way more efficiently than you could ever do with any other approaches we know about. More importantly, in some sense, in addition to sort of providing practical and theoretical leverage on just this assembly problem, it turns out they solve a wide variety of other problems to do with noise filtering for sequencing and resequencing of pretty much all of the data sets we've been able to look at. And so if you go to my blog post, I linked to some grant applications where we wrote up how to do this for things like resequencing for cancer and so on, to the point where I think we can throw away 80% of your data while retaining all of the actual signal in your data set. So hopefully I think, you know, if I have to make an overbold statement, I would just say the problem that comes from generating too much data from sequencers, from short-read sequencers, can be solved within a year or two at an engineering level with the approaches we've developed. And all of our source code's available, and I write blog posts and stuff about this stuff all the time, and I'm happy to chat more. But almost more important than that, the algorithms, the filtering algorithms we're developing are many core compatible and even pretty distributable. And we've been able to do things like use Amazon for a lot of assembly problems because we've decreased the memory footprint so much. So between those two things, we've been trying to provide a path forward to the next generation of computing architectures, which are going to be many-core or potentially GPU. I'm not so sure GPU will work, but also, because we've done this weird thing of making our preprints, our papers available via preprint archives, making our source code available with tutorials, before publication, there are already dozens of labs using digital normalization, which is the streaming lossy compression approach, and it seems to be working just fine. So, okay, so that concludes the hardish science section of my talk. And now, I'd just like to remind people I was actually asked to discuss this. So be nice. So here's some not so casual observations from a lifetime of interacting with physicists. And the phrase that Carol used on our phone call about this was, there's an intellectual fusion that has not taken place between biology and big data and biology and physicists who deal with big data and science every day. Why hasn't that happened? So I'm going to make some sweeping generalizations. But first, I don't hate physicists. Far from it. My father's a physicist. He's a nuclear theorist that some of you may know. My first Unix account was given to me by a guy named Mark Galassi, who apparently has attended several of these XLDBs. My first publication was with a physicist named Chris Adami, who's actually now 2 doors down from me. I went to grad school in biology partly because when I asked Hans Bethe what he would recommend for a young scientist, he said biology was where the most opportunities were, which is, you know, when people like Hans say that, you tend to listen. I've published a lot with Steve Koonin, who, again, I have to imagine a number of the people in the audience know. The shell model for the Monte Carlo, now I'm garbling it. But anyway, he's done a lot of computational science work. And then this is actually one of these weird things. Kurt Callan showed up at, who's the particle physicist, showed up at Caltech to do a sabbatical and wanted to play with some biology and had some great ideas. And I was actually one of the few biologists that would talk to him. And so we ended up publishing a very nice, my second favorite publication. My new favorite publication is actually from my lab, right? Because now I'm the senior author on that. But and it's a really great publication, and it gave me a chance to get to know Kurt. So overall, I'm very physicist positive as well as physics positive. And I think I've written a blog post on this that I link to again. And biologists don't like me saying this, but models play a very different role. So in physics, models are often predictive and constraining. So you can build a model, and based on sort of some abstract notion of aesthetics, and often it actually turns out to fit reality reasonably well. You need to measure some things to get initial conditions, but then you can actually go forth and use that model to predict fine-grained outcomes that are useful. And in biology, that just doesn't happen. Very rarely do you know enough to use modeling. When you do, you can apply it, you can look for sufficiency, but it doesn't mean the model is correct, it just means that it could be correct. And really it turns out that in biology, models are very rarely predictive of the actual observations. So things like evolution and natural selection tell you how things are going to happen and why, but not necessarily the specifics of what you're gonna see at the end of the day. And so in particular, in my graduate lab, we assembled, mostly other people assembled, this map of all the genetic interactions in the first 30 hours of sea urchin development. So it took about 15 years and probably 200 man-years of research. Only in the last 5 years or so have we been able to start using this model to say, yes, in theory, this process could be completely, the observations we make could be completely explained by the interactions that are on this map, right? Which is where you kind of want to go, right? For disease, you want to be able to say, yeah, when we look, we can explain what we see based on the model that we have. And that's extraordinarily rare in biology, as far as I can tell. So the thing that peeves me the most is that there's little or no tradition of computation in biology. And that's partly because there hasn't been much in the way of big data or modeling. I like this statement: ecological and evolutionary models are regarded with suspicion, guilty until proven innocent, right? And there's essentially zero computational training at the undergraduate and graduate levels. I like to say biologists often got into biology at the undergraduate level because they wanted to do science or med school and wanted to avoid math and computation as much as possible, right? And so this is now having a real poisoning effect. The senior PIs we have avoided this along the way. The classes we have, which are taught by the professors who are trained in this way, don't discuss computation or modeling at all, basically. And it's extraordinarily frustrating. worse for me, frankly, and this is all about me, is that there's really a sick culture of computation in biology. They want, first of all, if you want to develop a computational method, people say, yeah, but how do we know it works? How do we know it's interesting? That's a valid point, but they're becoming increasingly important. So you should probably respect them as independent scientific endeavors, and that's a hard road to hoe. And I had to go that road by becoming a biologist first, and then circling back around, which is not the fastest route between 2 points. Biologists really want push-button software that just works. If I hear one more person say, look, just give me the answer and get out of my way, I'm going to scream loudly. And my particular complaint right now is that evaluation and validation of software just by users, not by the authors, but by users is very rare. So people want a package that they can download, they can run, they get an answer, and then they go back to the lab. They don't want to think about whether or not the assumptions that went into that computational method, the assumptions that the parameters that they chose, they don't want to think about any of that. They don't want to know if those assumptions are correct. They just want to get an answer and move on. And so there's this famous statement in biology from about 20 or 30 years ago, a few days in the library can save you a few months in the lab, right? And here I really like to point out to biologists that a few weeks in front of a computer can save you a few years in the lab, so less snarky but more accurate, or just as accurate. So and the frustrating thing is with the culture problem, it's just hard for me to explain to biologists how bad this is, right? How much of biological research could be better if they spent more time evaluating hypotheses before they went into the lab to test them. So again, I can talk about that later. So biology is built on facts and not theory. So when I worked with Kurt, there was this experience. He came along with this optimization of DNA binding models in E. coli. And basically, no matter what, we couldn't take the 48 known CRPs and develop a model that would pick out those 48 and not also pick out 300 to 3,000 extra sites in the E. coli genome as being valid. And it turns out, the real breakthrough in that paper was, it turns out that these 300 to 3,000 extra sites are evolutionarily preserved in such a way that indicates that they are functional, that evolution cares about them. And Kurt said, how is that possible? And I pointed out that we don't actually know that much about E. coli, which might surprise a number of physicists, right? So those extra binding sites are almost certainly real and functional. But Kurt didn't realize how little knowledge we actually had about one of the central models of biology. And so he really, my contribution was actually just to point out that we didn't actually, that he was probably right. And then I actually had Mark Galassi sitting on my couch a week or two ago, and he said, I ride the bus up to Los Alamos every day, and I see these biologists and bioinformaticians typing away and doing sequence analysis, and it looks really interesting. Can I get involved without learning any biology? Right? And if you want to drive a biologist crazy, right? If you want to just push them off the edge, say this kind of thing in casual conversation, right? Biology is all about the details, and the more details, the better. If you want to get into biology, you have to love complexity and complications and data, because very often the best theory is going to be something that doesn't shave away all the details, but that includes most of those details. And so I think, and I make this point in this accompanying blog post, I think that physics is less about integration and more about developing a simple model, a simple mental model. And biology is more about integration of the data into a mental model. But again, I'm an amateur at this sociological stuff. So one point here is the reason that The Origin of Species, which is one of the central theoretical texts in biology, right, was such a success is because when you read it, he says, look, there's this very simple idea. And by the way, you can explain almost every observation you make about the natural world by appealing back to this original theory. But it doesn't tell you how exactly the natural world's going to be. It just tells you how to interpret it, which is a very sort of different, I feel like it's different. Okay, so I just want to point out my career path. I started working in math, and I did research on an evolution model and Earthshine observations of global albedo. And then I spent 11 years in learning biology, and now I'm doing all sorts of other things. And the main point I want to make is, if you want to work in biology, there might be shorter ways to do it, but I can guarantee if you spend 11 years getting a PhD in it, people will treat you like you're a biologist. Again, maybe not the fastest route, but I have no problem actually convincing convincing biologists that even though I'm doing primarily computational work at this point, that I'm a biologist, because I've logged my years at the bench doing pipetting. And I can explain why that's not going to get them very far in some ways. Anyway, okay, so some very simple concluding thoughts. Biologists don't trust models and data analysis approaches that come from outside biology, often because they're mismatches. I think that the skill set and attitude that physicists can bring to biological research is really important, but you have to sort of accept that most of your facts are wrong, are useless for biology. So, and unfortunately, for better or for worse, funding is on the side of biology, and you need to meet them halfway, or more than halfway. And my big real point is, gosh, I wish biologists would learn from the mistakes that physics has spent 30 years getting past, right, in terms of modeling and data analysis and data storage, software development, and it's just been very frustrating. So to watch biology repeat these mistakes. And that's all I have to say. I'm very happy to chat. It sounds like there will be beer and wine to chat over, which is good for the sociological stuff. And thanks for listening, and I'd be happy to take any questions.